we finished we finished last week with discussion of pattern pattern recognition receptors. You remember this picture? Extremely artistic. So today we talk about phagocytosis? No. So today we're going to talk about phagocytes, how they consume microbes. Very similar, yes. A little bit more details. So it happens that ape and micro have immunity at the same time, or like I have, huh? What? Really? Okay. I didn't notice. Anyway, phagocytosis. Those two cells. First of all, which two cells can perform phagocytosis? Macrophages. Oh, come on. And neutrophils, right? Macrophage. Let's say it's a macrophage. Recognizes the pathogen and forms pseudopodia around it. Looks like a Pac-Man. Okay, pseudopodia, false legs. So how macrophage, remind me how macrophage tells apart the bacterial cell and the normal cell? By recognizing what? Yes, pathogen-associated molecular patterns, PAMP. Okay, so it recognizes the cell, the bacterial cell, and engulfs it. And this bacterial cell is now in the vesicle that is called how? How is it called, this vesicle? If it's phagocytosis. Phagosome, yes. It's in the phagosome. Next to the phagosome is another vesicle, full of proteolytic enzymes, reactive oxygen species, and acid. How do we call this? Yes. Proteolytic enzymes and reactive oxygen species. This all is lysosome. Those vesicles fuse together and form phagolysosome. In phagolysosome, all those chemicals inside of the phagolysosome, acid, proteases, and hydrogen peroxide, for instance, they will break microbe in pieces. Does that make sense? So let's name this structure. It's phagolysosome. Okay, cool. And then, of course, the remnants of the bacteria are thrown out of the cell by the process of exocytosis. Is that clear? Okay. So far, which mechanisms of innate immunity we covered? We covered inflammation, it's a general mechanism. We covered phagocytosis, which is the component of inflammation, right? Because phagocytes migrate into the tissues and destroy bacterial cells and damaged cells. Next is the evolutionary ancient system that is called a complement. Okay? When I say evolutionary ancient, that means that we can find complement in such organisms as arthropods. Okay? And actually, how to say, it's a lot of immunologists consider complement in humans somewhat of an oddity. It's really a weird system. It's definitely a relic that we still have, and it still works against microbes. So, complement. It's a system, okay, a system of blood proteins that can be activated in a cascade fashion. If you remember coagulation pathway, especially intrinsic coagulation pathway, you may recall that the activation goes, factor 12 activates factor 11, factor 11 activates factor 9, factor 9 activates factor 10. Okay, so it's one activates another. Same is true for the complement. One protein activates another, which then activates the third, which then activates the fourth, and so on and so forth. That makes sense. Terrific. So there are like maybe a dozen of different proteins in a complement, and you don't have to know them. Okay, that's kind of a relief. But what you do have to know is what triggers the complement activation, and what are the outcomes of the complement activation. Triggers. Classical pathway. And the trigger going to be antibodies attached to a pathogen. Okay. Then you have lectin pathway, which is triggered by sugars, not just sugars. Man knows sugars on the pathogen surface. And the third one is alternative. Now, alternative pathway is triggered by molecules, certain molecules that can be found on the surface of some microbes. Does that make sense? Are we clear on the triggers? Okay. Interesting thing about complement. So each trigger initiates the chain of events, chain of activation that all comes down to the same convergence point, C3b factor. And C3b then initiates a number of responses, and we will highlight what are the responses. Number one is so-called opsonization. Which means that, let me try and illustrate it. So imagine that this red is the bacterial cell. Okay? In opsonization, complement proteins, which I'm gonna make green, okay? They're gonna stick to the surface of the bacteria and make it more attractive for phagocytes. Does that make sense? Okay? So essentially, opsonization will increase phagocytosis of the bacteria. Another way is to generally, quite non-specifically, increase inflammation. right? The third way is to form membrane attack complex. Which, you're not going to believe it, attacks the membrane. Yes. So if this is the cell, membrane attack complex, it's what I think it's C5, C6, C7, C8, and C9 proteins. I never remember them. I never worked on the complement, so I'm not gonna ask this, but why? But I want you to know the idea. So membrane attack complex is when you have a bunch of proteins that form a pore, okay? MAC, okay? They form a pore in the envelope of the bacteria, and then fluids like cytoplasm and things start to move around in the bacterial cell, out of bacterial cell, and bacteria essentially dies. Does that make sense? Mm-hmm.
On the slides, this says they all converge first at C3 on your slide.
Oh, C3, C3b, you don't have to. It's fine. It's fine. Okay. This is what you have to know. You have to know the things that induce it and things that come out of it. Does that make sense? This is what I will ask. Maybe not everything, but I will ask. We're clear. One of my favorite components of immune response, innate immunity, are the interferons. These are the proteins that can be of two types. Type 1 and type 2. They differ, type 1 and type 2. We're not going to go into how they differ, but they functionally slightly different. Okay, type 1 and type 2 interferons. What's fascinating about them is the way they're produced, the way they signal. How does that work? Imagine that you have a cell and you have a cell next to it. Okay, and then you have some distant cell. And each cell, of course, has a nucleus. Okay, and the first cell gets infected. So this infection is sensed by the cell. Using what? What's innate mechanism for sensing the microbe? Innate mechanism that senses the pathogen. We call them pathogen. So sensed by pattern recognition receptors, which sense pathogen-associated molecular patterns. That makes sense? So sensed by PRR. Those are receptors, and they trigger the intracellular signaling pathway. And that signaling, the signal goes into the nucleus. What is in the nucleus? There you go. DNA. And DNA consists of what? The informational units in the DNA. How do we call them? Genes. So genes that are responsible for the interferon production are expressed. Does that make sense? interferon gene expression. And then interferon is released from the cell. And what surrounds the cell? Cells are bathing in what? Extracellular fluid. So interferon now is an extracellular fluid. And then interferon can signal. What can it signal to? No, they don't have a receptor. All other cells. Okay. Can interferon, now for signaling on all other cells, it has to interact with something on the surface of the cell. With what? Receptor. Again, receptor. Can receptor be on the same cell? Yeah. Yes, so that's going to be autocrine signaling. Can it be on a different cell? The adjacent one, like next door. Paracrine signaling. Or interferon can go into blood and get delivered to some distant cell. Does it make sense for you? Okay. And that's going to be endocrine signaling. Now, using that adjacent cell as an example, when interferon, okay, let's say this black thing is interferon, interacts with its receptor, what is going to happen next? Receptor is going to trigger something. It's going to start something. What receptors usually start? Eventually, yes, but they deliver something inside of the cell. They start something inside of the cell. In order to get it started, you have to deliver the what to the nucleus? Signal, okay? So they start the signaling pathway, okay? Interferon receptor will start the signaling pathway that will reach the nucleus and initiate gene expression. Does it make sense? I want you, by now, you have to be really comfortable with the idea that everything that happens in response to infection, during the infection, it's all about gene expression, protein synthesis, and alterations in the gene expression will determine the response and whatnot. Does that make sense? And then the cell, when it initiates this gene expression, those so-called interferon-stimulated genes, ISGs, that's not going to be an exam. Interferon-stimulated genes can make cell refractory to the infection. If interferon signals on the infected cell, this interferon may initiate apoptosis. Cell will die, and whatever's inside of the cell will also die. Does it make sense? It may crank up the synthesis of enzymes that destroy RNA of the virus, for instance. Does it make sense? Okay? So, essentially, interferon is the warning system. Infected cell produces interferon to warn other cells about the infection, to give them time to get prepared. Does that make sense? It's like, I bring up zombie apocalypse often. So if I was bitten by a zombie, and I know that I'm going to turn into a zombie in like 20 minutes, first I want to tell you, run for your life. And then I want to go on the PA around the college and tell everybody else, run for your life. Does that make sense? So paracrine signaling is to the next door neighbors, and endocrine signaling is to everybody else. Why pay so much attention to interference? Because they're curious, first of all, they're interesting. And they determine a lot of immunological differences between the species. I bet everybody heard about Zika virus. It's a flavivirus infection that's transmitted by mosquitoes, and in men generally it causes fairly mild fever with retroorbital pain and some joint pain, and then it is gone. In men it was shown to be able to infect testes, so it potentially can be transmitted sexually, although not high frequency. But in pregnant women, apparently the virus can cross placenta and cause brain malformation in children, in fetus, leading to the microcephaly, intrauterine growth retardation, and other pretty nasty effects. That makes sense? So if you take a model pregnant woman, which of course we're not going to do, and inoculate this model pregnant woman with the Zika virus, there's a great likelihood that she will give birth to a child with a microcephaly or other brain abnormalities. If you take a mouse, pregnant mouse, and inoculate it with the Zika virus, nothing's going to happen to a fetus. If you take another pregnant mouse, which does not have receptors for interferon, does that make sense? So there's no interferon signaling, and inoculate this pregnant mouse with a Zika virus, you are going to see intrauterine growth retardation, symptoms that are similar to human microcephaly, and so on and so forth. So there's something about mouse interferon that protects mice against Zika virus. It's true not only for Zika virus, it's also true for yellow fever and somewhat true for dengue, you know. So there's a great, actually, mystery. We don't really know what's the difference. We don't know the mechanism, why mice are protected. We know that if you take interferon away, they become a decent model for mosquito-transmitted infections. Other than that, no idea. So, interferon is pretty effective in treating diseases. Well, should be, apparently. Yes?
If interferons are created by the DNA, wouldn't it be something in their DNA that would make it? So they weren't infected by malaria?
Say it again.
If interferons are created by the DNA in the cell,
They encoded, yes.
encoded,
Yes.
then wouldn't the encoding for that interferon only be in the mouse DNA and not ours?
There are differences in sequence between mouse interferon and human interferon, right? But here's the trick. They don't remove interferon. They remove the receptor. Does that make sense? They remove the receptor. So they're completely disrupting all production of all interferon-stimulated genes. Does that make sense? And that creates a lot of uncertainty. If you want to compare interferons, there are going to be differences, but they're going to be limited. If you talk about tens or maybe dozens of interferon-induced genes, you don't really know what kinds of differences we talk about. Does that make sense? Because there are a lot of them. Okay? Now, interferon is, as you can see, pretty effective against virus infections. Can we use it to treat people? Can we use interferon to treat people? Yeah, we can. But the problem is that it causes a lot of side effects, and of them, sort of minor, like there is constipation, nausea, and gastrointestinal symptoms. But probably the biggest one is depression. And suicidal tendencies. So, use of interferon to treat long-term infections is kind of limited. It is used to treat infections that have no specific therapy, like yellow fever or dengue or Ebola or, I don't know, Hunin virus. There are plenty of infections that we have no specific drugs against. But up to a certain point, hepatitis C, acute hepatitis C, was treated with interferon. Well, not anymore, because Gilead Pharmaceuticals recently released the drug that cures 99.9% of infected people. So interferon is a drug, but it's definitely not a good drug. Okay? Does that make sense? So understand how this thing works. Okay? Infected cells produce interferon, and then uninfected cells respond to interferon. Okay? With that, we conclude the innate immunity and move on to adaptive, which is more complicated but also more exciting. What do you need for adaptive immunity to develop? Exposure to what? Pathogen. Okay. So it doesn't, cells that mediate adaptive immunity, you already have them. But in order to build up the response, the cells have to encounter a specific pathogen. Okay. So you need exposure. Time frame. It's slower than innate. Okay. And location-wise, like innate immunity, often, we talk about the localized infection, like a bruise, okay, or cut, or a bump, or something like that. Local injury. In a systemic response, it's, okay, adaptive response is systemic, Hunter's right, okay? Which means that cells that mediate it go in the blood, and the molecules or cells that actually respond to the infection, they reach every corner of the body. Does that make sense? Also, imagine that you had mumps. Are you going to get it second time? Okay, mumps is not chickenpox. That's the only disease that you still know. If you had chickenpox once, you're going to get it second time. No. It's not mutated. Shingles is different. So if you actually had chickenpox, the virus goes latent and stays in your neurons. And you get shingles when you get older or immunocompromised. It's the same virus that you're infected with. Okay? But if you get, say you get chickenpox at age of 10, you can kiss the person with chickenpox, you can hug, you can do whatever you want. You're not going to get infected second time. You're not going to get that pustular rash all over your face. No. Because now you have an immunity. What is that feature? How can we call that feature? When memory, immunological memory, that's the critical feature of adaptive immunity. Think about this. If you think about model situations, so you have innate immunity, okay, and you expose your innate immunity to a microbe A. Every time your innate immunity will encounter microbe A, its response is going to be more or less the same in terms of the time frame, in terms of intensity. So you expose your adaptive immunity to microbe A, it builds up the memory, second, third, fourth, fifth exposure will be much faster, much stronger than the first one. Does that make sense? Because now you remember the microbe. Okay? Does that make sense? The memory concept. Okay. Which cells do we need for adaptive immunity? Lymphocytes. And there are two types of them. T and B. Where do they start? They start in the bone marrow. Okay. And where do they go next? It's not funny. Where do they go next? On the bone marrow. They naive. They're not ready to fight yet. They have to mature. Where do they mature? That's why we call one of them B cells, huh? Formally, yes, but they go to special organs. So one of them, huh? Okay, T cells go to the thymus. Okay. It's not heart, it's a thymus. It's pretty much how it is shaped. Okay. Really? Huh? And bone marrow, the B cells, they stay in the bone marrow. Okay. And they mature there, and we're going to talk about the process of maturation. And once they are mature, where do they go? The lymphoid organs, secondary lymphoid organs, namely spleen. Did you ever see spleen? So it looks like this. Spleen and lymph nodes. Trust me, it's a bean-shaped. It is a bean-shaped. Well, no, it's okay. Huh? No. No, it's fine. But spleen, it's probably proportion-wise, it should be a little longer. It's like long and flat organ. I can vouch for mice. I took a lot of spleens out of them. Okay, so they seed secondary lymphoid organs. And what do they wait there? They wait for something, which comes after the exposure. They wait for the exposure because, think about this, the extracellular fluid constantly drains into the lymphatic system, taking with it all the pathogens, all the microbes that are in tissue. Okay, so when they, after the exposure, where do they go? In the blood. They go into the blood, and in the blood, they start to do whatever they're supposed to. T cells are responsible for the branch of immunity that we call cellular immunity. And this branch is mediated by cells. Okay? So T cells, after the exposure, when they get activated, they will become so-called T effector cells. There are different types of T effector cells, which we will have to chance to talk about. Now, B cells are responsible for humoral immunity. What's the humor? Huh? Fluid. Huh? It's not the bone, and it's not. There's nothing funny about it. It's the fluid. In ancient Greece and ancient Rome, doctors believed that people had 4 humors: blood, bile, phlegm, and something else. Huh? What? I don't remember. I think they had 4, then somebody else got added 5th, and they believed that all diseases were about the imbalance between the humors. So like if person gets really apoplectic, like really red, you know how they treated the potential stroke? No. They let blood out. They decrease the blood pressure essentially by letting the blood out, by bleeding a person. Okay? So kind of trying to bring humors back into balance. Which is of course utter and complete bullshit, but they didn't know better. We can't really judge them. So humoral means that this component of immunity is something that is soluble in fluids, which turns out to be antibodies. So B effector cells, which we're going to talk about as well, produce antibodies. Is that clear? That's kind of an overview of what the innate immunity, adaptive immunity, sorry, is. Now we talk about exposure so often. Exposure. What these cells have to be exposed to. The general name, antigen. You don't have to expose to pathogen technically. What can be an antigen? Aspirin? Huh? Yes, but what, I mean, can you name, can you, say, huh? Then it's not going to respond, that we're not going to respond if we don't have receptors. Something that we should have receptors for. But, Shauna, expand. Something foreign, but what foreign? You know what I'm trying to say? What, can you think of anything like biological molecule that can be an antigen? Huh? Protein, okay, good enough. No, Sean actually brought a very important point. It should be something foreign. You know, we'll get to the explanation why it's not always true. But protein, what else? DNA. RNA. Keep going. What other biological molecules you know? Large, complex, which, so that's the general characteristics. Large and complex. What other large and complex molecules you know? Huh? Amino acids are small, and we'll talk about how to make antibodies against them. A little small, but many different sugars stuck together, polysaccharides, complex polysaccharides, sure enough. Okay? Lipids actually can be. You see, the general characteristics of all those molecules is that they are large and complex. Can you think of a molecule that is large but not complex, like extremely large? It's going to be a polymer. Like the one they make globes from, nitrile globes. It's a polymer, which is essentially a molecule that consists of many rather simple fragments. And these fragments are identical. So it's not, it has no chemical complexity. Does that make sense? It's sizable, I mean it's huge, but it's not complex. So it's large and simple, not an antigen. Does that make sense? Can you think of a molecule that is rather small, but complex? Not like huge, enormous. What about, I don't know, somebody proposed amino acid. Let's say amino acid. Bless you. Small and complex. So the question is, if you will take amino acid and stick it into the mouse, will mouse produce immune response to it? No, it doesn't satisfy the condition of being large. But can you make it seem bigger? You can. How you can make something look bigger? Huh? In this case, you will alternate the chemical structure. You have to stick them together and it's going to be completely different chemical. Yes, you have to attach it to something bigger. Think about monster trucks. Like, monster truck is really big, okay? But if we will take the large wheels, it's going to be much smaller, right? So that happens here. If you take that small molecule and stick it to the large carrier, which is usually a protein, then this will become an antigen, okay? An immune system may develop the immune response against that small molecule that is called haptin. Does it make sense? Haptin is the little small molecule that attaches to the carrier, can be attached to the carrier, okay? And then immune system can recognize it. Now, it all may sound very irrelevant, but think about, so poison ivy. Usually when you're exposed to the poison ivy for the first time, you don't even know about that. What happens is the chemicals, the organic chemicals present in the poison ivy, enter the bloodstream and attach themselves to your own proteins. And your immune system recognizes them and produces the immune response against them. Usually on the first exposure, the immune response is minimal, but what's more important, your immune system creates memory. Does that make sense? When you get exposed to the poison ivy the second time, those same small molecules are now recognized by antibodies, T cells, and so on and so forth, and you get the full-blown dermatitis. That is result of the immune response. It's not that poison ivy is toxic or something. It's your immune system that responds to it. Some people are refractory to poison ivy. Does that make sense? Okay. Now, say antigen is a protein. Proteins consist of what? Amino acids, yes. And say it's a protein, 200 amino acids. So your immune system will not produce antibodies, will not produce response to the entire protein or to each individual amino acid. It will recognize and produce the response to so-called antigenic determinants. So if this is the hypothetical protein, this is the antigenic determinant 1, this is the antigenic determinant 2, and this is 3, which doesn't mean that there's going to be only 3, you know. There's several antigenic determinants on the molecule. Think about this. you have a child or you have a, I don't know, brother, sister, whatever, there are some body features that let you to recognize the person. I mean, show me a patch, just patch of the skin from, you know, fragment, photograph of the skin from my son's back. I would be able to tell that this is him because it looks like any other skin. But people have little like birthmarks, scars, I don't know, moles, eye color, very distinctive features that allow us to recognize them, okay? That makes sense? This is antigenic determinant, essentially. So each antigen has certain structural features that stand out, that immune system can recognize. Make sense? Now, to show you a better example of it, imagine that you have a protein like this. Okay? Which has 3 antigenic determinants. You can fragment this protein, okay, so that you're going to have this antigenic determinant separated. It's a fragment of a protein. It's like, I don't know, 10, 15 amino acids. You can stick that antigenic determinant into the mouse and create immune response to it. Okay, immune response. And then say antibodies. You can take antibodies to that antigenic determinant and see that those antibodies will recognize this entire protein. Does it make sense? If you take a fragment that has nothing to do with antigenic determination, like this fragment, okay, and stick it into the mouse, and then isolate antibodies, they will not recognize the original protein. Does that make sense? Did I make it clear? It's antigenic determinants, the fragments of the antigen that your immune system recognizes. Okay? Now, I mentioned several occasions I used the word antibody. Next, I want to talk to you about what it is and the structure of it. Now, I'm going to call it antibody, but in fact, antibodies, T-cell receptors, B-cell receptors, they share similar structure. Does it make sense? So they're all pretty similar. Antibodies, B-cell receptors, and T-cell receptors. So what is that structure? Antibody has 2 fragments and 4 chains. Okay, so this is the typical structure of antibody. It's a fork with 2 prongs and a handle. They taught me the word prong yesterday. I didn't know it. 15 years in the States, I didn't know the word prong on the fork. So my AP2 class taught me the word prong. I never had a necessity to name them in my life. It's a fork. I use it for eating. I never had to use. I never had to name those things. So yesterday was the first time, and it taught me it's prongs or tines. T-I-N-E-S. I would call them needles. Or teeth.
Oh yeah, I've heard.
Okay. Anyway, so it's a fork. It has a handle, which is black part, okay? And it has two prongs, which are red parts. Got it? So the handle, the black part, is called FC fragment. C stands for constant. And the red part, well, actually two parts, called FAB fragments, which stands for antigen binding. Okay? You can also appreciate that antibody consists of 4 chains. Okay? 2 light chains and 2 heavy chains. Does that make sense? And 2 heavy chains. Now, which part would functionally recognize antigen, bind to the antigen? What do you think? Antigen binding. The prongs, right? And constant part would be constant. Now, so antigen binding part, it should be really different, correct? It cannot be the same for every antigen, because then there would be no differentiation. So each antibody is unique. It recognizes its own antigen. It's like a key, okay? Does that make sense? So each key will recognize its own lock. So how can antibodies be so numerous, so variable? Essentially, this is the antibody is the protein. Proteins, where do they come from? Where's the information about proteins stored? In the DNA. In the form of what? Each protein is what? A single gene. No, I mean gene. Single gene, okay? So one gene for a protein. Now, antibodies, the protein, there should be a gene for each antibody, right? So how many antigens are out there? What do you think? Well, give me a number. A billion? Trillion. Let's go with a trillion. Okay, it's bigger. A trillion. Okay, trillion antigens. It's actually quite realistic. How many species of bacteria? We know probably 100,000. Each has hundreds of different proteins. Okay. Add to it strains, add to it viruses, add to it all the animal species and plant species, all antigens, right? So our immune system has to be able to produce antibodies to each and every one of them. Okay, so trillion antigens. Let's say it's a protein. Let's say it's a protein of 100 amino acids. How many nucleotides do we need to encode one amino acid? 3. Right? One codon is 3 nucleotides. So to encode 100 amino acid protein, which is a small protein, we need at least 300 nucleotides. To encode trillion proteins like this, we need 300 trillion nucleotides, which is 100,000 more than there is in a human genome. So obviously, having a gene for every possible antibody isn't working. How? Oh, we know. We know pretty well. It's pretty cool, actually. The answer, simple answer is the license plate. The complicated license is VDJ recombination. which sounds really fancy, but it's actually pretty straightforward. So imagine that there's a fragment of human genome that contains 3 types of genes. V gene, well, genes, V genes, D genes, and J. Okay, J and D. Say you have, well, on this picture you have 1 J, 3 D, and 5 V genes. Are you with me? Okay. No, it turns out 4, okay? 1, 2, 3, and 1. So, the antigen binding region of the antibody consists of 3 individual genes, 1V, 1D, and 1J. Does that make sense? Give me a number from 1 to 4. Okay. From 1 to 3, 1, and here's 1. So this is one combination. Another combination can be, I don't know, 4 to 1. Okay, 1, 3, 1. Does that make sense? There are different combinations of V, D, and J genes. And each combination of 3 will pretty much make an FAB fragment. There are, first of all, there are more, not only VDJ, there are some more genes like C genes and something else, okay? And I think it's 47 V's, 12 D's, and 21 J. Some numbers, okay? Pretty big numbers. Now, if you combine those numbers, if you will try to calculate how many combinations those genes can produce, turns out the number is almost infinite. Do you understand the point of recombination here? It's like a license plate. You have 26, you have 10 numbers and 26 letters. But the combinations that you can make out of it are almost endless. Does that make sense? So same happens here. And well, we'll get to the B cells. So pretty much every version of antibody, every combination of V, D, and J genes will have a property to bind one specific antigen without binding anything else, because they're going to be structurally different. Does that make sense? Different sequence, different structure. Does that make sense? Okay, great. Now, since we got this introduced, let's talk about B cells. I want to remind you that, they produced in the bone marrow, stay to mature in the bone marrow, and then they go into the secondary lymphoid organs and wait for the exposure. First of all, what is B cell maturation? Which answer B cells have to answer to? What should B cells, using the B cell receptor, what do they have to recognize? B cells. What do they have to recognize? Huh? So in immunological terms, it's antigen, yes. They have to recognize an antigen. Surface protein of a virus, is that an antigen? Sure. I don't know. Porin proteins in the outer membrane of gram-negative bacteria, are they antigens? Yes. Albumin, human albumin, protein in the human blood, is that an antigen? Why? It's antigen. But it's still an antigen. But you got the right point. It's not foreign. So which antigens B cells have to recognize? Only foreign ones. Okay? So they pretty much asked a question. Not only it has to recognize foreign antigens and respond to them. Does that make sense? So it has to be able to tell apart. We can formulate this question as response to foreign antigens only. Does that make sense? Now, if it does respond only to foreign antigens, then is that a good B cell? It's perfect. What if the answer is no, and it responds not only to the foreign, but also to autoimmunity? What do we have to do with that cell? Kill it. Apoptosis. Does that make sense? Now these guys that are good functional B cells, where do they go? After the maturation from the bone marrow. No. After the maturation, secondary lymphatic organs, okay? Secondary lymphoid organs. Spleen and lymph nodes, okay? B cells, mostly lymph nodes, right? And they wait for the exposure. So imagine that you have I'm going to make it colorful. So you have 3 populations of B cells. These fork-shaped molecules are B cell receptors, okay? And each population is called a clone. Now, these 3 clones, you have billions of clones of B cells, encounter the antigen. Okay, so it's antigen. Which clone will recognize the antigen? The red one, yeah. I mean, that's why I color-coded it. So there will be no recognition here, no recognition here. Now this guy will recognize, there will be recognition, okay? And that will lead to 2 things. Cells going to differentiate, and they're going to proliferate. What does differentiation mean? Cells become more specialized. Does that make sense? Functionally specialized. Proliferation. Multiply. You get more of them. So functionally specialized. What 2 types of cells B cells will produce? One is memory B cells. Another is plasma cells. Plasma cells will actually go into the blood and will start to churn out antibodies. Does it make sense? So plasma cells will start to produce antibodies. Do they have to be of any specific color according to my scheme? Which color? They have to be red, right? So this specific clone of B cells, we're going to call it red clone, will produce red antibodies. Don't take color thing literally, okay? Does that make sense? And these red antibodies will recognize the antigen. Okay? Now, antibodies. Can I move on? Is that good? Antibodies. What major types of antibodies do we know? We know 5. Wait a minute. Don't. I will give you a chance. Go ahead. Wait. IgG. Next. IgM. IgE. Sorry. IgA. Yes. What is the most boring one? No. M's pretty neat. IgD. Well, it's subjective. Come on. If you think IgM is boring, who am I to tell you that they're not? IgD. No. By what they do. IgD is the B cell receptor. Okay? IgD. It's a B cell receptor. That's its main function. Okay? Which are the evil ones? Well, from my personal standpoint, they definitely are evil ones. IgE. Yeah, because of the allergies. So IgE causes degranulation. Degranulation. What happened to my writing? Degranulation of mast cells and basophils. Normally, that will increase inflammatory response, okay? But in many cases, like in my case, it will lead to the allergic reaction. Okay. We're going to find IgA. If they are secretory antibodies. Mucus secretions. Actually, serous secretions as well. So like tears, snot, saliva, sweat, breast milk. The morning class, it's such a trouble figuring out what is the secretion that supports the life of other people. They proposed saliva, tears, and sweat. But eventually they got the breast milk. Okay? So think about this. For 6 months, the breastfed children are protected for the entire period of breastfeeding from the by mother's immunoglobulins A. Okay? So that's one of the benefits of breastfeeding. IgM are the first responders. Which means they're produced first during the primary exposure, right? They also form this gigantic agglomerates, the 5 antibodies stuck together, like a snowflake shaped. And finally, IgG, they're most numerous. They carry out a lot of, they can do pretty much everything, okay? An important feature of IgGs is that they can cross placenta. Now the pressing question is how they work. There's several ways antibodies can work. One is that they can opsonize the pathogen. We already talked about opsonization. Now in this case, imagine that this is the bacterial cell, okay? And antibodies will just stick to it, okay? Does that make sense? That's opsonization. Why opsonize? What is the... Remember we talked about complement. Opsonization increased something. Huh? Phagocytosis. It makes microbes more attractive to phagocytes. Correct? Okay, so that's opsonization. What else they can do? They can agglutinate. What's agglutination? Clumping of cells. So if, say, this 3 cells and antibodies will bind and form some kind of a clump. Okay, first of all, they will restrict the mobility of potential pathogen, right? Second important thing, this big clump becomes more, how to say, prone to phagocytosis, more noticeable to phagocytes. Does that make sense? Third mechanism is precipitation. And this is, it's not essential, but pretty much the same as agglutination, but it works not for the cells, but for the molecules. Okay, so you have a bunch of molecules. What can be a molecule that we can build the immune response to? What can be a molecule that we can build the immune response to? Bacteria is not a molecule, but some of them produce those molecules. They release them outside and they kill us, those molecules. Toxins, yes, toxins. They produce toxins. Venom, the snake venom. Do you know how anti-venom serum is produced? They say, we talk about copperhead, okay? They keep copperheads in captivity. I saw a fantastic video of a guy who actually watches the colony of copperheads. I never saw a more calm person. Like he pulls out, not copperheads, sorry, cobras. Pulls out the cobra for feeding, okay? Pulls out the drawer and throws the rat into the drawer. Cobra is agitated, so the cobra climbs out of the drawer and falls on the floor. This guy absolutely just no panic. He just picks it up, puts it back. And the cobra falls out. He puts it back. So calm, like he does it every day, for like I don't know how many years. So yeah, so these snakes are kept in the captivity, and then you can collect the venom either by using the glass beaker. Okay, you can pretty much make snake to bite a glass beaker, and the venom starts to pour down the beaker. Or you can also use some electrical current applied to the venomous glands. That will produce more and faster. So you collect venom and you inject it into a horse. Small doses. So horse is not going to die, but the horse is going to produce antibodies. Then you take a little bit of the blood, isolate antibodies, and you get the horse immunoglobulin, the shot. And it's anti-venom serum. Does that make sense? Oh yeah, it is used on humans. Probably because they're big. You can take blood like fairly easily. Okay. Also, when they consider the production of antibodies, it's also about cross-reactivity. So if horse immunoglobulins, will they recognize human antibodies and stuff like that? Human proteins and stuff like that. Okay. So fourth mechanism is neutralization. This is mostly about viruses. It works in a fairly simple fashion. Imagine that you have a virus, okay? Antibodies bind to it. Now, in order to infect the cell, virus has to bind to something on the cell surface. What it is? The receptor. If the virus is all surrounded by antibodies, can it bind the receptor? No, it can't. So it's kind of stuck here. It can't infect. That makes sense? And fifth one that we kind of discussed, activation of complement. Neat? Remember classical pathway? Good. Those are 5 mechanisms. Let's take a break, and it looks like we on the good